All right, I'd like to thank the organizers for giving me the opportunity to speak today. My name is Sarah Reiff, and I'll be talking about kinases and Stentor. So we've already gotten a little bit of background about Stentor, but I just want to reiterate that it is quite a large cell. It's about 1 millimeter long, and the cortex is also highly polarized and highly differentiated. So for those of you who missed the beginning of Tatiana's talk, at the posterior end, we have a holdfast that the stentor can use to attach to surfaces. And then at the anterior end, we have the oral apparatus that's ringed by a membranellar band that consists of sheets of cilia. And then along the length of the cell, we have rows of cilia. But one of the most interesting aspects of Stentor is its ability to fully regenerate after being cut in half in a way that perfectly preserves the original cell polarity and morphology. So ciliates in general tend to have some limited regeneration capability, but Stentor is a good model for regeneration, partially because of its extensive regeneration ability, and also its large size allows us to cut the cells easily and observe them under the microscope. So regeneration is one of the long-standing mysteries of biology, but there are still a lot of questions remaining unanswered. So we still don't know how the cell really can establish and maintain polarity during regeneration, or when the cell creates new structures, how does it position them into the right place and scale them to the size of the cell? And then finally, of all the events that go on during regeneration, how are they coordinated globally throughout such a large cell? So we do know that kinases are really important for a lot of the processes that go on in regeneration, such as centriole biogenesis, ciliogenesis, and cortical organization. So for example, the PLKs are really important in centriole duplication in other systems. We know that NEKs are important both in the cell cycle and in ciliogenesis. There are marked kinases that are important for microtubule organization, and it's really well established how CDK expression helps regulate the cell cycle. So in general, the question I'm interested in answering is what the kinome of Stentor can tell us about the signaling capabilities of such a large cell. So I've been involved in the draft genome of Stentor, which has been quite a big collaborative project. So right now our genome size is about 83 megabases, which is a bit smaller than we were expecting, but we've identified nearly 35,000 protein coding genes, which is quite a lot of genes. And then one of the interesting things we found in the genome project is that the Stentor's genes tend to use the standard genetic code, which is different from all the other ciliates whose genomes have been sequenced so far. And we also found that the genome is very intron poor, and 82% of genes lack introns altogether. Of the introns that we do find, they tend to be 15 to 16 base pairs, which as far as I know is the smallest intron size in any organism known. And our collaborator, Estienne, has also found introns of the size in condylostoma, which is related to Stentor. So I've used hammer and blast to take all the protein coding genes and look for kinase domains. And what I find is that Stentor encodes over 2,000 protein kinases, which is a total of 6% of all Stentor's protein coding genes. So this is a huge complement compared to metazoa and yeast, and even plants. But then when we look at kinomes of other ciliates, Stentor's numbers are kind of in keeping with Paramecium and Tetrahymena. So I've classified all of Stentor's kinases into families and subfamilies, and we find that there are a lot of kinase families that have been quite expanded in Stentor. So there's a few families like the histidine kinases, which were reported in Tetrahymena and Paramecium to be highly expanded, that are much less so in Stentor. But then we also have families like the CDPK family over here, which is a calcium-dependent protein kinase family that was originally identified in Arabidopsis, which has 257 members in Stentor. And one of the other things we find is that there are a lot of mitotic kinase families like the NDRs, the CDKs, the PLKs, NEKs, and Aurora kinases, which are quite expanded in Stentor, especially compared to human and yeast. And just as a side note, I'll mention that paramecium has expansions in a lot of these families as well, but we can keep in mind that paramecium has undergone a couple whole genome duplications, so their total number of genes is higher compared to Stentor. But this idea that the mitotic kinase families are expanded is interesting to us because previous scientists have noted a lot of parallels between regeneration and cell division in Stentor. So in these two figures side by side just kind of we can compare the way that the oral apparatus is created, first in cell division for the posterior daughter cell and oral regeneration after the oral apparatus is removed. So we're really interested in the hypothesis that regeneration might proceed in part by the cell activating cell division pathways at will. But one of the other things we noticed in terms of kinase families expansions is this family called the DIRCs, which is actually a dual specificity tyrosine kinase, which is highly expanded in Stentor even compared to other ciliates. And this is a little bit interesting because, as some of us heard in Ron Perlman's talk yesterday, the other ciliates lack canonical tyrosine kinases of the type that are found in metazoa. So I was interested in looking into the idea of tyrosine phosphorylation in Stentor a little bit more. And it turns out in metazoa, there are also other serine threonine kinases that have been studied and found to have additional tyrosine phosphorylation ability. So a few of the examples of this include WEE1, which phosphorylates CDK1 to regulate the cell cycle, and then ST7 family kinases, which generally act as MAP kinases, and then CK1, which actually has a variety of functions throughout the cell and phosphorylates a variety of targets. But one of the more recently described targets was the histone 2A, in which it phosphorylates on a tyrosine residue. So we looked for all of these targets in Stentor and performed alignments of all the Stentor orthologs. And we found that in the case of Stentor CDK1 and histone 2A, we find greater than 90% conservation of the phosphorylated tyrosine residue from metazoan systems. And then in the case of MAP kinases, the phosphorylated tyrosine is conserved in more than 2/3 of the MAP kinases. So this points to the suggestion that Stentor does indeed have tyrosine phosphorylation ability. But back to the idea that we have these expansions in a lot of kinase families, it raises the question of whether all these families are redundant in function or whether some of them have diversified and undergone functional specialization. So we looked at other protein domains present on kinases, and we find quite a number of other domains that are present on Stentor kinases. So there are some domains such as EF hands, which are used to bind calcium ions, which we find in quite a lot of Stentor kinases, and nucleotide binding domains, which we expect to find. But then there are also a number of other protein domains that are present in only a couple examples of Stentor kinases. And a lot of these are serve functions like protein-protein interactions or structural functions or targeting to specific areas of the cell. And we actually find a couple examples of novel kinase domain architectures that haven't been described before. And one of the greatest examples of this is a case of CDPK kinase that has a couple adenylate kinase domains at the N terminus, and then a few partial domains in between. So at this point, we have no idea what this protein does or what its function in the cell is, but we could, speculate that, for instance, it could represent a coupling of calcium signaling to energy metabolism in the cell. And then we found a second example of a Pac-like kinase that has the 5 domain at the N terminus. So 5 domains are typically involved in targeting proteins to the endosomal membrane. And 5 domains have been found on kinases before, but as far as I know, not a kinase of this specific family. So it looks like it could be a novel gene fusion, and perhaps represents a new specialization of a protein in this family to the secretory pathway. So in conclusion, we find that Stentor encodes a much greater complement of kinases than most other organisms outside the ciliates. And we find that mitotic kinase families in particular are highly expanded. So we're really interested in exploring whether cell division proteins are involved in things like regeneration. And we also find indirect evidence for tyrosine phosphorylation despite lack of canonical tyrosine kinases. And we're really interested in doing like Western phosphotyrosine to look for more direct evidence of tyrosine phosphorylation. And we found a couple examples of novel domain architectures that point towards Stentor-specific functionalization. So what I'm working on now is an RNAi screen of the kinases to look for ones that are specifically involved in regeneration, and primarily focusing on mitotic kinase families. And I'll mention that our grad student Athena is also helping with this and focusing on the auroras specifically. And Tatiana mentioned that our previous grad student, Mark, had found that the kinase regulator MOB1, which is part of the mitosis exit network in yeast, was found to be an important polarity marker in regeneration. And then recently, I've done some knockdowns on homolog of CDC2 and found cell division phenotype. So we're really interested in doing regeneration experiments on these to see whether regeneration is affected. So I'd like to thank everyone in the Marshall Lab and all of our collaborators, and the people you see on here with starred names are the people who were heavily involved in the Stentor Genome Project. And then we're funded by the NIH. So thanks for listening, and let me know if you have any questions.
Hi. Do you have any evidence for different expression transcript levels in growth, division, and regeneration for some of these kinases?
Not yet. That's definitely something we'd like to do. We have some RNA-seq data, so we can see that we can see expression levels like just during vegetative growth, but we don't have that data yet.
So did you find that when you were looking for within the genome, were the kinase domains pretty conserved, or would you not pick that up with your searches? And also, did you find ones that were outside of those conserved families? Or again, would those not be picked up?
Can you repeat the question?
So what I'm asking is how divergent were you, did you explore the edges of the divergence of the
Okay, yeah. So there's definitely a spectrum of conservation in the kinase domains, and Stentor does have lots of kinases that we couldn't classify into families because they're too divergent. Additionally, we also looked at the number of pseudokinases that we see in this, and we find about 16% of the total kinases look like pseudokinases, and that's more highly represented in the unclassified kinases.
And what was your proportion of unclassified? Just a guess.
I think it's close to half.
Have you tried a pharmacological approach, seeing if any kinase inhibitors affect regeneration?
So we have tried that, and we found that lots of kinase inhibitors at their normal concentration metazoan systems seem to not affect Stentor. But we've also found this for, I guess, other types of drugs in general. So it just seems as though either Stentor has some way of like pumping out drugs really effectively or not taking them up. But yeah, we have tried that, but thanks. All right.